Hey science students, last time we took notes, we talked about meiosis and mitosis. Remember that mitosis happens from my head to mitosis, but meiosis happens in my gonads, okay? Meiosis is gonna happen in the testes for males to create sperm, which have half the chromosome number of 23 for the sex gametes, for the sex cell that is passed along to the offspring. Meiosis happens in the gonads for females, which are called ovaries, to create eggs, which have 23 chromosomes as well, which is half the number of a normal cell, or a somatic cell, I should say. And so that egg has 23 chromosomes, and 23 chromosomes in mom's egg plus 23 chromosomes in dad's sperm makes a total of 46. And when they first join, we need to add this. When they first join, so get your pencil, that first joining is called a zygote. Okay, I forgot to say that. The first cell, zygote equals first cell. So in when the sperm is swimming and it gets to the egg, it fertilizes the egg, that is called a zygote. The first joining of the sperm and the egg is a zygote. Then the zygote will split, okay? It'll go through mitosis, okay? It will divide and make an exact copy of itself. Okay, and that 2 becomes 4, and the 4 becomes 8, and the 8 becomes 16, and they keep dividing, okay? They are called embryonic stem cells, and embryonic stem cells keep making more of themselves, and they have the ability to be any kind of cell in the human body, okay? All of the DNA can be activated and become any kind of cell for a human. Eventually those cells get differentiated. Some of them become the spinal cord, some of them become the brain, some of them become the heart, some of them become skin cells, some of them become like stomach lining cells, and so they start to differentiate, they start to become different, and once they differentiate, they can only be that kind of cell. At that point, the DNA is turned on for the things that that cell needs to do. Okay, so the first cell, remember, is called a zygote. And the reason why we want to remember that is it'll help you understand some of the terms we're going to go over today. Okay, so first term that we are going to go over today is alleles, okay? It's a hard one to say, allele. You have to kind of get your mouth like back like that, allele, okay? All right, so an allele, we're going to write up here. I'm just going to, you might use a new page. A-L-L-E-L-E, okay? Allele. And then that equals different forms of a gene. Okay, so here, down here we have a gene. So maybe we should, well, I'm gonna redraw it because you guys might have it on a separate page. So there's our DNA. And the DNA has these, it looks kind of like a twisted ladder is what a lot of people call it. It's a double helix is the correct term. On the DNA, there will be a spot, and that spot will code for a gene, okay? So you're gonna have several genes on your DNA, and different spots on the DNA code for different genes. There's actually a lot of spots that don't code for anything. It's just like kind of left over from evolutions, and actually viruses have put a lot of their DNA within our own as we've evolved over the years. So anyway, the genes are the things that are the important part of the DNA that's going to code for your traits. Okay. Now you might have an allele from your mom that's going to code for hair. Okay. So let's write an example. Okay. So let's say you have. Let's just take one kind of example of curly hair or straight hair, okay? All right, so hair is what we're talking about. That would be the trait, okay? So the trait would be hair. And then the allele could be curly or straight. STR, straight. So those are the two kinds of alleles. Okay, now if we choose for one of them to be dominant, okay, a dominant trait is going to be the one that's going to dominate over the other trait. So there could be a dominant allele equals curly hair, and the recessive comes from the word recede or stand back. Recessive allele would be straight hair. And I don't know this for sure. I'm just making these up right now. Okay, I don't know if curly hair is actually dominant or recessive. But we're going to start with this. Okay, now we need to give it a letter. Okay, and I'm going to choose H for hair, and the capital H is going to represent. Capital always represents the dominant. So in parentheses, I'm going to put a capital H right here, and I'm going to put a lowercase h by the straight. Okay. So if you were to get a dominant from your mom and a dominant from your dad, then you have the same two alleles, okay? And so same is also known as homo, okay? That's I think they call it a morpheme, and that morpheme, homo, means same, okay? And then we're going to talk about zygote. So we're going to say zygous. Or I guess you could say zygote too. Homo, so as an adjective it would be zygous. So homozygous dominant, meaning that you have two dominant genes. So that equals homozygous dominant. What do you think that would represent? We have two lowercase that are the same. Same. Homo. Zygous, talking about the first meeting of sperm and egg. Homozygous. Recessive. And then our last option is one of each, okay? And so we're going to use a morpheme that means different. Hetero. Heterozygous. Oh, and I guess you just say heterozygous. You could say heterozygous for curly hair because it's the dominant, but it's heterozygous. You have one of each, okay? And these are known as genotypes. Genotypes, because they are the three types of genes, okay? So So you have this type of gene, you have this, or three possible combinations of genes. So these are the genotypes. Okay, and it's the genes that you get, the actual genes. You might not know what all your genotypes are. You probably know what your phenotypes are. Like you could say, I have curly hair. If you say you have curly hair, then you could be homozygous dominant, but you could also be a heterozygote. Okay, you could have a straight gene in there that's being hidden. Okay, so that's one of the main things that Gregor Mendel found out. Okay, so let's we get the alleles, okay, which are the different kinds of traits that you can get. You can get dominant alleles and you can get recessive alleles, and then those can be shown in three different ways: a homozygous dominant, a homozygous recessive, and a heterozygous. And those three things are the three genotypes. Those are the three possible genotypes. Okay. Now, let's say genotypes equals possible combinations of genes from parents. And then one more word over here is the phenotype. Phenotype is how you look. How, or what is being expressed. How, so it's not always how you look, it could be how you smell. It could be how your emotions are. So there's how you look. It really should be what is expressed. What is expressed, what is seen, or what is noticed. So phenotype is how it ends up being, how it ends up looking. Okay, and then I'm gonna kind of make a new page over here. And we're gonna say that, so Mendel, he was the one that kind of figured out dominant and recessive traits. And what he did was he would take green peas and mate them with green peas and mate them with green peas and mate them with green peas until he knew that the only possible thing that he would ever get would be green peas. And that's the purebred, okay? The purebred, like homozygous dominant or homozygous recessive is what he wanted to get. He wanted to get parents that he knew the only thing that they had was that one trait to be passed on, okay? So this time I'm gonna go with the letter G, and we're gonna say that that's dominant green, and the lowercase g is gonna be yellow. You usually use the same letter for the allele. Sometimes I might use like C, but the letter C, it looks the same as a capital as it does a lowercase, so I like the letter G a little better because it's got a different look to it and you can see it easier. So what first generation yep, you can see that. First generation, Mendel, crossed. So you're gonna, oh, this is called a punnet square, okay? And you wanna draw this punnet square. Now he didn't figure out the punnet squares until later when he analyzed his data and he realized the percentages, and then he started to figure out how the punnet squares worked. Actually, he didn't do the punnet squares. A guy named Punnet did. But the percentages helped Gregor get to the point where these things make sense, but it's easier for us to understand them by starting here. So he would take a green pea, and like I said, it was purebred, meaning it only had green. So I'm going to put a capital G up here and capital G up here, and I'm just going to say that this is going to be the male. You don't have to do that. I like to separate my Punnett square by putting that extra line there. Not everybody does that. And then on the female side, I'm going to do a lowercase g and a lowercase g. Okay? So here we have one parent's genotype, and here you have another parent's genotype. This parent's genotype is homozygous dominant. This parent's genotype is homozygous recessive. So I have a purebred green and I have a purebred yellow. Okay, now what I need to do, what Mendel did, was he bred the purebred green and so I bring this capital letter G down, and I also bring it down to this box, and then I bring this capital letter G down, and I also bring it down to this box. And this is something you need to practice. You'll get a lot of practice at this, hopefully, and you'll get better at it. And just completing a Punnett square, it's pretty basic. You just bring these capital letter G's down. This one goes down to here, this one goes down to here, this one goes down to here, this one goes down to here. Now I do the same thing for the lowercase letter G's, but I bring them across. Okay, so I bring this over here. And I bring this one all the way over here. And I bring this one over here. And I bring this one all the way over here. Okay, so you can see that the offspring, these are all the possibilities of what the offspring could get. And they're all the same. Okay, so all of the offspring would have a capital G and a lowercase g. So they got one of each allele, so they got different alleles, and so that means that they're heterozygous, and they're going to be green because they got a capital G that's dominant, and so they're going to end up being green. And they're going to be carrying, they have a hidden gene or a hidden allele that's not seen. That recessive yellow allele is not seen, and they have that in their genetics. So now, in order to see if it was still there, the way that Gregor Mendel figured out that it was still there was that he took two, so I'm going to say second generation now, second generation. He took two that were from the first generation. He took two of these offspring and he plugged them in. And he mated them so that he took one of them and took the pollen from one, which is similar to pollen's like sperm. It's the male gamete. And then he fertilized the egg of another one to make, so he took a heterozygous parent that was male, the pollen, basically, the pollen, and then he took he fertilized the egg of a female heterozygote. Now you'll see this one looks a little different. Try to do it on your own if you can. Bring the capital letter G down. capital letter G down. This time we're bringing a lowercase letter g down. And we're bringing a lowercase letter g down. And now we're going to bring this one has a capital to bring across to here. And you're going to bring it across to here. And then we bring the lowercase letter g across to here. And the lowercase across to here. Now you can see we have different combinations that are possible. Okay, so one of the possible combinations is this capital G, which is homozygous dominant, and that's going to be 1 out of 4, which equals 25%. So he didn't always get 1 out of 4. They didn't always just have 4, but you know, if there was, it was always 1/4 or 25%. Okay, so if you had 100, 25 of the 100 would end up being homozygous dominant. About. Okay, it's a probability. It doesn't always happen perfectly, but it happened over and over and over again with lots and lots and lots of plants with lots and lots and lots of characteristics. And he noticed that about 25% of the time he would end up with homozygous dominant. I mean, he really couldn't. Actually, what he did notice, hold on. 2 of the 4, this one and this one, are heterozygous. Heterozygous. 2 out of 4 equals 50%. Okay, so now I can say what he really did notice was the 75%. Okay, so 50 plus 25 is 75. So it's 3 out of 4, or 75%, ended up being green. And so he was able to notice the phenotype, how it looked. Okay, how it looked. The phenotype he was able to notice in the end. And then, this is what he really realized here, is that lowercase, homozygous, recessive, 1 out of 4 equals 25%. So he noticed that the yellow came back, okay? Here, in the first generation, the yellow disappears. None of the babies had turned out yellow. They all ended up green because they all have a dominant gene. But in the second generation, because each of the parents, the male has a possibility of passing on the lowercase recessive yellow gene. The female has a possibility of passing on the lowercase recessive gene. And so 1 out of 4 of the times, the offspring is going to get the yellow gene from both parents, and therefore the recessive gene is the one that is expressed because there's no dominant gene hiding it. And so that's what he noticed, is that the yellow gene came back. So here's the yellow parent. None of the offspring have that kind of gene or that kind of look. None of the offspring are yellow. But then in the second generation, the yellow comes back. And that's how he realized it. The recessive was being hidden by the dominant gene. Okay? All right. That's the notes for today. You guys have a great day.